NCERT Exemplar 12 Principles of Inheritance and Variations. First question, all genes located on the same chromosome form different groups depending upon the relative distance, form one linkage group, will not form any linkage groups, form interactive groups that affect the genotype. So all genes located on the same chromosome, if you say this is one of the chromosome, if all genes are located on one chromosome, this is the chromosome, homologous pair of chromosome, whatever the genes located on one chromosome is called linked genes. So that form a linkage group, that is going to form a linkage group. So what is the answer here? That form one linkage group. Conditions of karyotype 2n plus 1 and 2n plus 2 are called as 2n in human being is said to be 46 chromosomes. 2n plus 1, it might be a Klinefelter syndrome or it might be a Down syndrome, and 2n minus 1, that means 45 chromosomes, it might be a Turner syndrome. So all this, what do you say? Aneuploidy. We call that the feature as Aneuploidy. So what is the answer for this? 2n minus 1 or 2n plus 1 is said to be Aneuploidy. Okay, moving on. Distance between the genes and percentage of recombination shows. Distance between the genes and percentage of recombination shows a direct relationship, an inverse relationship, a parallel relationship, no relationship. Distance between the genes and percentage of recombination shows a direct relationship, that it shows the recombination frequency. If a genetic disease is transferred from phenotypically normal but carrier female to only some of the male progeny, the disease is transferred from phenotypically normal but carrier female to phenotypically normal but carrier. It is possible in case of hemophilia. Some of the male progeny, in this case only one of the male progeny, 50% probability to get their son, her son is going to be hemophilia or color blindness. So in that way we can say this is sex linked recessive, this is sex linked recessive here, or you may call directly it as a X linked recessive. Answer is sex linked or X linked recessive character, hemophilia or color blindness. In sickle cell anemia, glutamic acid replaced by valine. Which one of the following triplets code for valine? GGG, AAG, GAA, GUG. So what is our answer here? GUG, GUG. A person having genotype IA-IB would show the blood group as AB. This is because pleiotropy, codominance, segregation, incomplete dominance. Person having genotypic IA-IB, that means here a person with A blood group married a person with B blood group, we get both A and B. Okay, so that it can be explained like this. A person is having A sugar polymer and the RBC crossed with B sugar polymer and the RBC, that this offspring will have both A sugar polymer and B sugar polymer. We call this feature as codominance. This feature is called codominance. ZW type of sex determination is seen in. ZW type of sex determination is seen in platypus, snails, cockroach, peacock. Such kind of sex determination you will find in birds, so in that way I am selecting peacock. A cross between two tall plants resulted in offspring having few dwarf plants. What would be the genotype of both the parents? Okay. So in the first cross, capital T and capital D small t, we get only tall plants, and here 50% tall and 50% dwarf will come. Here all tall will come, and in this case a cross between two tall plants. So this is that two tall plants, which we can say both tall plants are heterozygous. There you get 75% tall plants and 25% dwarf plants. In a dihybrid cross, if you get 9:3:3:1 ratio, it denotes that alleles of two genes interacting with each other. It is a multigenic inheritance. It is a case of multiple allelism. The alleles of two genes are segregating independently. In the Mendel dihybrid cross, the taken two characters, seed color and seed shape, they are present and located on two different homologous pair of chromosomes, like this, capital Y, small y, and this is capital R, small r. So we call it as they are present on two different homologous pair of chromosome. Hence, the alleles of the two genes at the time of meiosis, they get segregated independently. They get segregated independently. So what is our answer here? They are independently assortated. Which of the following will not result in variations among siblings? Independent assortment of the genes, crossing over, linkage, mutation. Okay, if a particular gene is located on same chromosome and that is very closely linked each other, if the two genes are very closely linked, we can say that pattern is linkage. Even at the time of crossing over, you will find this particular two genes collectively move towards the next generation. So what do you say? Which of the following will not result in the variation among siblings? If the genes are linked, the two genes are present, which are present very closely linked on one chromosome, they collectively transfer to the next generation. Mendel's law of independent assortment holds good for genes situated on the non-homologous chromosomes, homologous chromosomes, extranuclear genetic element, same chromosome. What is same chromosome? If the two genes present on the same chromosome, we can call the pattern as linkage. Okay, then Mendel laws of independent assortment to be possible means the selected two characters like seed color and seed shape of the Mendel should present on two different homologous pair of chromosomes. This is one homologous pair and this is another. Then only independent assortation is possible. So, what is our answer here? Mendel's law of independent assortment hold for good genes situated on non-homologous. This chromosome is homologous to this one and this chromosome is homologous to this one. We call these two are non-homologous chromosomes. So, what is our answer? Non-homologous chromosomes. Occasionally, a single gene may express more than one effect. The phenomenon is called as multiple allelism, mosaicism, pleiotropy, polygyny. One gene affects many characters, we say pleiotropy. The feature we call pleiotropy. In a certain taxon of insects, some have 17 chromosome and other have 18 chromosome. The 17 and 18 chromosome bearing organisms are. Okay, so this is little controversial here. We have two types of methods in this case, XX-XO method and ZO-ZZ method. In XX-XO method, if it is female, that is having 18 and male is having 17 chromosome. In ZO-ZZ method, female is having 17 chromosome and male is having 18. So, in these two cases, it might be male females respectively, female males respectively might be, but that NCERT exemplar taken consideration that some have 17 chromosome, other have 18 chromosome. 17 and 18 chromosomes bearing organisms, they are given male and female. So, might be they are taken consideration of XX and XO method. And then they said it as insects. That's why most insects are having XX and XO method. In that way, we selected this particular answer. The inheritance pattern of a gene over generation among humans is studied by the pedigree analysis. Character studied in the pedigree analysis is equivalent to quantitative trait, Mendelian trait, polygenic trait, maternal traits. So, generally for hemophilia, like genetic disorders like hemophilia, color blindness, sickle cell anemia, for these cases we do pedigree analysis, and all these are collectively considered as Mendelian disorders or Mendelian traits. Single gene disorders we consider as Mendelian traits. It is said to that Mendel proposed that the factor controlling any character is discrete and independent. This proportion was based on the results of F3 generation of a cross, observations that the offspring of a cross made between the plants having two contrasting characters shows only one character without any blending, self-pollination of F1 offspring, cross-pollination of parental generation. So, what is our answer here? Observations that the offspring of a cross made between the plants having two contrasting characters shows only one character without any blending. Two genes A and B are linked. In a dihybrid cross involving these two genes, the F1 heterozygote is crossed with homozygous recessive parental type small a, small b. What would be the ratio of offspring in the next generation? So, if it is like Mendel that selected two characters capital A, small a, capital B, small b, that might be on different chromosome, you may get a ratio of 1:1:1:1. But in the problem they said both are present on same chromosome. Both are present on same chromosome. So, definitely we can expect a single ratio here like 1:1. Linked in a dihybrid cross involving these two genes, F1 heterozygote crossed with homozygous recessive. Means here the cross you have to say like this, capital A, capital B, small b, cross small a, small b. If it is Mendel, you may get this kind of ratio. What is Mendel? Selected two characters on different homologous pair of chromosomes, but now they said they are linked. They are linked, that's why I selected that we get only two varieties, 1:1, because both the characters are linked. Both the characters are linked. In the F2 generation of a Mendelian dihybrid cross, the number of phenotypes and genotypes are, in the F2 generation of a Mendelian dihybrid cross, what is the phenotypic ratio of Mendel? 9:3:3:1. What is the genotypic ratio? 1:2:2:4 to 1:2:1:2:1. So, in that way we will find 4 plus 5, 9 here genotypes and 4 phenotypes. So, what is our answer here? 4 phenotypes and 9 genotypes. Here we have 4 phenotypes and 9 genotypes. Mother and father of a person with O blood group have A and B blood group respectively. What would be the genotype of both mother and father? In simple format we can say like this, mother and father should be both heterozygous only to get along with A, B and AB, that an offspring O will come here. So, in other terms we can show like this in genotype in genes format. Let me show you, because that is 2024 NEET question even, that I will show you in the genes format. Let me erase this. In simple format we can show easily to understand. We wrote like that. It is like this, IA I cross with IB I. Okay, so when IA I means O blood group, that you get like this, small I. So, in that format or in this format, we can show easily here. Okay. So, that means homozygous, heterozygous. In the heterozygous format only, you get like this. Let me write once again, IAI, IBI. So, this is the parental genotype. So, or you may call in simple terms like this, AO and BO. That answer is mother is heterozygous and father is also heterozygous. So, where do we find that answer? Mother is homozygous for A blood group, father is heterozygous for B. It is given in first option. Mother is heterozygous for A blood group and father is homozygous. This is given in second option. Both mother and father are heterozygous for A and B blood group respectively. This is our answer. That's it. Principles of inheritance and variations, exemplar problems. Thank you.